Hello, welcome to the next in the series of review quiz for the Biology Theory 201 public examination. In this clip, we'll continue our examination of cell reproduction, specifically dealing with the specific type of cell reproduction known as meiosis. By definition, meiosis is a type of cell division that results in gamete, that being sperm and egg, being created with half the number of chromosomes, the haploid, as the parent cell. The purpose of meiosis, the reason it exists, is to do two things. One, to ensure genetic continuity, and as well to assist in, to promote genetic variability. As a process, meiosis occurs in the gonads, testes in males, ovaries in females. And there are two types of meiosis: oogenesis, which results in egg production, and spermatogenesis, which is the production of sperm. Just as with mitosis, meiosis occurs in a series of stages and steps as well. Meiosis occurs in two distinct stages, the first of which being Meiosis One. The second stage is Meiosis Two. Looking at Meiosis One specifically, it has four stages: Prophase 1, Metaphase 1, Anaphase 1, and Telophase 1. Meiosis 2 as well has four stages: Prophase 2, Metaphase 2, Anaphase 2, and Telophase 2. Looking at Meiosis 1 first, Meiosis 1 is also known as reduction division. Reduction division is the process of reducing the number of chromosomes in a cell by 1/2. In short, going from the diploid state, 2n, to the haploid state, n. In the case of humans, every one of our body cells contains 46 chromosomes. After Meiosis 1, the cells that are produced contain 23. The first stage of Meiosis 1 is Prophase 1. During this stage, homologous chromosomes, that being similar chromosomes, line up next to each other as pairs. These pairs are known as tetrads. And at times during this stage, during Prophase 1, a process called crossing over of non-sister chromatids will occur. When these chromatids cross over, this results in an exchange of genes. In other words, it increases variety. It increases the possibility of genetic combinations. As you can clearly see, during Prophase 1, a diagram which you see here, the chromosomes are aligned in terms of tetrads, and each tetrad consists of a pair of homologous chromosomes. Crossing over, as occurs during Prophase 1, is when the sister chromatids, as aligned, actually cross over each other and exchange sections. Metaphase 1, the second stage of Meiosis 1, involves homologous chromosomes aligning in the middle of the cell, and the spindle from one end of the cell will attach to a pair of sister chromatids, while a spindle from the other end attaches to the other pair of sister chromatids. As you can see in the diagram, the tetrads are aligned, and the chromatids are as well aligned to opposite sides, going to be pulled in opposite directions by the spindle. Anaphase 1 follows Metaphase 1. And during Anaphase 1, the spindle pulls the homologous chromosomes to separate ends of the cell. The diagram here clearly illustrates that the chromatids are being pulled to opposite ends of the cell. By doing so, we're actually pulling the sister chromatids in different directions. Telophase 1 involves the reappearing of the nuclear envelope around each set of separated chromosomes. The spindle will disappear. Cytokinesis, as you recall, this is division of the cytoplasm, occurs. And two new cells are created with, and this is key, half or haploid the chromosome number of the original cell. Beginning with 46 double-stranded chromosomes, each new cell now contains 23 double-stranded chromosomes. Or from the 96 chromatids that we began with, each cell now contains 46 chromatids. The end result being a pair of cells, as we see here. Following Meiosis One, Meiosis Two now begins. The stages of Meiosis Two are identical to the stages of mitosis. Prophase 2, the first stage, we see the following events occurring. The nuclear membrane appears. The spindle forms, and the chromosomes become visible. Next, metaphase 2 will begin. Here, the chromosomes line up at the middle of the cell, and the spindle attaches to the centromere of each chromosome. With Anaphase 2, the spindle pulls a single chromatid from each chromosome to opposite ends of the cell. And Telophase 2, we see the following. The nuclear membrane reappearing around each group of chromatids, and they are now single-stranded chromosomes. The spindle disappears. Cytokinesis occurs. And two new cells are created, each of which has the haploid number of chromosomes. Previous public examinations have asked questions on this very topic. An example which I provide here. Where does crossing over occur? Crossing over occurs, remember, with the chromosomes, the homologous chromosomes align. They exchange sections. This occurs during prophase 1 of meiosis. Another example question that you may expect to see. When does separation of the homologous chromosomes occur? Again, we are looking at the homologous chromosomes as opposed to chromatids. This would not be fertilization, as that is when gametes fuse. In mitosis, the homologous chromosomes do not separate. This leaves with Meiosis 1 or Meiosis 2. In Meiosis 2, we have separation of the chromatids. The correct answer then is B, Meiosis 1. It is during Meiosis 1 that the homologous chromosomes separate from each other. And this question, an error in which stage of meiosis would produce a human egg cell with 24 chromosomes? Again, you're looking at the separation of the chromosomes. The correct answer to this would be Anaphase 1. Thank you very much for your time and your patience. I hope this learning clip is helpful for you.